Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. So today we're going to be talking about reprogramming cellular identity in the brain, and we're joined by a special guest, Sydney Leaman, who's a PhD student at the Centre for Neurodevelopmental Disorders and the Centre for Developmental Neurobiology at King's College London. Sydney holds a bachelor's in human and medical genetics, a medical degree, as well as master's degrees in both genetics and neurodevelopmental disorders, and is currently completing his PhD in the lab of Benedict Berninger, where he is working on reprogramming cellular identity in the brain and more specifically in the neocortex. Thank you so much for joining us today, Sydney.
No worries. So many degrees.
Yes, you said it.
Yeah, it's a long list.
Very long. So with all your wisdom, we hope that you can enlighten us today about what reprogramming cellular identity is all about, and the challenges and the future and everything that you know about it.
Sounds fun.
So why don't we start by you telling us a bit about what led you to this niche of research interests?
So I think it's quite difficult to explain exactly where my interest in genetics came from, because it's been with me for so long. Like even in primary school, I was interested in genetics, and when they were cloning Dolly the sheep, that was like very exciting. When I was still just about to start secondary school, and yeah, I've really kind of been interested in that genetics ever since. And so that led to me taking on my genetics degree as my bachelor's, and so I studied medicine, as you introduced. That is, when you're young and you don't know, you're kind of good at science, and people tell you to do medicine. I don't come from a big medical family, so I kind of went into that a bit blind. And in the end, it's genetics, which was always my passion, and that kind of came through in my master's and in my bachelor's. And so really the interesting thing for me, the holy grail of medicine, is to be able to fully repair any tissue. So any tissue that goes wrong, if we could completely restore and repair all those tissues, we could treat an enormous amount. And genetics is a super powerful way, first of all, to understand the pathology of disorders, but then how our genetics can lead through development to very complicated disorders, such as those that affect the brain. And also as a salvation, in the sense that if you can specifically manipulate gene expression, you might be able to do interesting things with cells and harness their potential for medical purposes. So I think the way that I came to reprogramming was really just combining my passion for genetics and trying to achieve this like holy grail in medicine of trying to use genetics to completely regenerate tissue.
Yeah, I really like that, like combining your two passions. I didn't actually realize that before, because I think it's such a hot topic in neuroscience that I never really question why people are interested in it, but that makes total sense.
Yeah, I was wondering, but where does your niche interest specifically in neurons and glia lie? Because perhaps tissue reprogramming or cellular reprogramming that don't have anything to do with neuronal cells, perhaps like any other cell in the body.
Right. Well, so, I mean, all very interesting. So, I mean, the mammalian brain is particularly devoid of regenerative capacity. So, I mean, effectively, there are some exceptions, but pretty much all of the neurons in our brain stop dividing in utero, so before we're even born. So when we're born, we have all of the neurons that we're ever going to have. So that means that when they die or they're damaged, so if you have a stroke or you have any sort of injury or you have any sort of cortical damage or neurodegeneration, those neurons don't come back in any form. So, I mean, that's quite different to some other organs which do have stem cells and they can regenerate themselves, but most solid organs in the mammal, including the brain, don't have that. And so neurological diseases can be pretty devastating. And so there's that. But I wouldn't say that I'm so exclusively, I think there's so much that we can learn about regeneration in the brain from other tissues. So I don't think there's a very clear-cut divide. I think I have broad interests in regeneration in other tissues as well. But I think the disorders of the brain are particularly interesting to me. I'm also particularly interested in, I think partly through one of the things about doing medicine is that it doesn't really matter which specialty you're in, you come across psychiatry because it's just so pervasive, and it's so obvious when you're a medical student how little we both know about these disorders, but also our ability to provide any sort of treatment for those disorders. So I think from a clinical motivated point of view, I find that very interesting. And I think we're so far off understanding those, though, that I don't think regenerative medicine is the place where that will be the first school of thought. I think the more practical places where I think we can make real progress are during injury and during ischemia.
Mm-hmm.
But I guess that's a little bit of where my interest in the brain comes from.
Yeah, that makes sense. Basically, it's one of the most challenging, so why not try to
Exactly.
figure it out?
Yeah, that's what I was thinking. You picked the hardest problem and you wanted to get at it.
It's got some unique challenges, the brain. So of all of our solid organs, the liver is quite interesting because if you can pretty much take away most of your liver and it will just restore its mass and restore its all the functions that it does, all its homeostatic and synthetic functions that the liver does. But the brain, the thing about the liver is it's really quite simple. Like it's just a lot of, I mean, I'm being facetious, but it's a lot of cells that just need to come back and do a function. Whereas in the brain, you've got this exquisite wiring that needs to happen and is forged over your lifetime, all of these synaptic connections. So it's a bit more of a complicated task to think about how we regenerate it.
Yeah, and like we actually had an episode on neurogenesis with Carlotta, and in that I also mentioned whether or not neurogenesis occurs, the main challenge is also making sure that those newborn neurons are able to integrate and become functional, right? So that's always like the main issue with the brain, I think. And like you said, all neurons are generated like while we're in the womb, but then so much plasticity still occurs around birth, like when we're first born and stuff like that. And those processes are really important as well. So yeah, it's good that you highlighted that as a challenge. And it also leads me to my next question, which is, so if we're trying to regenerate neurons after trauma or ischemia or stroke or anything, if we want neurons, why are we trying to reprogram other cells in the brain into neurons instead of just, like manually adding neurons?
Yeah. So the first thing is I guess for listeners, I guess you guys know, but we can now, since the Yamanaka won his Nobel Prize for discovering that you can reprogram somatic cells that are fully differentiated back to an induced pluripotent state by overexpressing a cocktail of transcription factors, which essentially unwrite some of the epigenomic marks and rejuvenate those cells back to a state where they are proliferative and they can have the potential to be then differentiate into pretty much any cell type in the body, or at least most of them. So people are using those induced pluripotent cells, differentiating those cells into neurons in a dish or even in 3D culture, but usually in a 2D culture in a dish, and then you can transplant them into a place. And they do, to some extent, integrate and form some synaptic connections. And in fact, this has already been done. So in Parkinson's, they've shown in mice that you can transplant dopaminergic progenitors into the mouse midbrain, and you can restore some function in Parkinson's models of mice. And those have actually moved to clinical trials. So people are doing this. So I think that's one of the ways we can think about regenerating the brain, and that is being done. So the process, I think we can imagine a future where you take a patient's cells who needs them, you de-differentiate those, and then you transplant them back into their brains. And that is going to, that would be a dream. If you could get that to work, that would be a dream. In reality, it looks like what they're having to do just to standardize the medicine, because the whole process is a bit variable. So as things often are in biology. So what they're doing for these clinical trials is they're de-differentiating cells that don't belong to that patient, making the neurons they need, which go through quality checks in big batches, freezing them, and then those therapies. But it means that the person receiving them has to be on immune suppressants, which has, costs for that patient. So it's looking like those sorts of therapies, at least for the time being, will be very reliant on immunosuppressant drugs, although I do think that I hope it will be ultimately possible to do it from a patient's own cells if we can figure out some of the manufacturing things. So I guess what's different about, I guess we haven't explained what my PhD is about or what goes on in my lab, but we're looking at a slightly different approach, which is to convert the cells that are actually already in situ at the site of an injury, whether we can convert those directly into neurons without having to do this costly process of taking them out, de-differentiating them, ensuring they have variability, and then transplanting them back into a patient, possibly with the needs of immune suppressants. So it's we're trying to find ways to get cells that are endogenously already in the site of injury, that are already the patient's own cells, to become new neurons as an alternative. Who knows which one might be the more practical for patients. It's hard to know at this stage because both technologies are quite early.
Yeah. So a few questions. So firstly, I don't think we've explicitly said what the alternative cells to neurons would be.
We have not, no.
Do you want to?
Yeah, so in the brain you have neurons, but you also have other non-neuronal cells.
Wow, so much suspense leading up to this one word. I cannot wait.
Yes, so you have glia, so they are like the main macroglia are astrocytes and oligodendrocyte precursor cells. So those have some interesting properties that make them almost a bit stem cell-like. So for example, OPC, so if I can just shorten that, so oligodendrocyte precursor cells, they proliferate throughout adult life, and if you deplete them, they will replace themselves in the cortex. They form like a they are spatially arranged. They have territories in the cortex, and if you deplete them, they will just repopulate. So they are, and they also can differentiate into oligodendrocytes, and so they're a true kind of a unipotent stem cell population that already exists in our cortex. And then you have astrocytes, which they vary transcriptionally throughout the brain, but they are transcriptionally quite similar to neural stem cells. So neural stem cells, which exist, it's a bit more controversial in humans. I mean, there is an ongoing debate about whether humans have adult neurogenesis, like if there's ongoing neurogenesis in the adult. Certainly in the mouse there is, and there are some specific regions where neural stem cells exist, and astrocytes are transcriptomically quite similar. So both of those cells have attributes that make them quite interesting as potential cellular sources for generating new neurons in the cortex. And so our lab tries to exploit these properties by expressing transcription factors that will try to drive these stem cell-like cells that we have in our cortex towards a neuronal fate. That's what the whole purpose of the lab is trying to accomplish. And we do that because we think that one day they might be useful for adding new neurons to a circuit of the patient's own cells in situ at the site of injury for repair of brain circuits.
Yeah. So you're targeting astrocytes?
Yeah.
And with the expression of certain transcription factors and making them turn into different types of neurons.
Yeah, so exactly. So we actually target both of those two types of cells, so astrocytes and oligodendrocyte precursor cells, OPCs. It looks so one of the things that I've shown in my PhD is that actually it's the astrocytes that make the neurons. So the neurons that we're making actually come from astrocytes. That was a question that's always been a bit unclear about whether they come from astrocytes or OPCs. Turns out looks like they're going to come only from astrocytes.
Okay, well, firstly, that's remarkable that you can make neurons from glia. But I also know that there's a lot of controversy about whether this is what people are actually doing, because experimentally, you show that you've converted them into neurons by showing those cells express certain neuronal genes, essentially, right? Do you want to talk a bit about the controversy in the field? I know there's been such a long history.
No, well, it's quite exciting, really. But it's exciting for the sort of niche field that we exist in. I mean, it's interesting to scientists. I don't know how much general interest that is, but I mean, one of the things in science, of course, is debating the methods that you're using and whether sometimes the devil is in the
details.
Yeah, and this is like you're trying to be tricked. So there's been a lot of papers published claiming that you can convert astrocytes into neurons. And the reason why it's tricky is because the way that we express our transcription factors is using viral tools, usually. In fact, that's what all of the papers pretty much doing. You can do it with small molecule drugs, but mostly people are using viruses specific proteins. And so the way that the scientists know if the virus is in a cell is if by using fluorescent reporters like a red fluorescent reporter or a green fluorescent reporter. And what people were observing was they were using some tools that specifically expressed where they saw the fluorescent reporter only in astrocytes at the beginning of the experiment. And then some weeks later, they'd look again and they'd see that these fluorescent proteins were only in neurons. And their interpretation of that data was that the astrocytes had become neurons. But I've done a lot of work in my PhD, and there's other labs who've also done a lot of work to show that actually what's really happening is the astrocytes are dying, and as they die, neurons, for reasons we don't understand about this specific virus that some of these experiments used, were taking up the fluorescent reporter.
Yeah.
Interesting.
So, yeah, interesting.
I mean, it's really unfortunate, but it's also highlighted, probably that's an understatement, but it's also highlighted the problems of using these viruses, which, people, I don't think really realized before. And now everyone is so much more aware of it.
Definitely, yeah. So, I mean, there's loads of stuff we don't... So the viruses they're using are obviously hugely common tools in neuroscience. I don't know how much you've talked about AAVs or
I don't think we ever have,
No,
really.
yeah, we should do an episode on that.
Yeah.
So, I mean, viral tools for genes... Viruses are amazing, right? So viruses have evolved over millions of years to be excellent at infecting cells. That's what they do. And they either insert their genetic cargo into the genome or they sit somewhere in the nucleus and they're expressing. So they're hijacking the cell's own cellular machinery to express genes. And scientists have very cleverly figured out ways to make sure that these take out the viral genes, which would be harmful because they usually involve replicating the virus, which damages the cells. So they've taken out all those genes and just put in the genes that they want expressed. And this is the way that modern neuroscience operates. And so none of neuroscience is possible really without the genetic and molecular tools that have been developed to express genes that we want in specific cell types. Mm-hmm. That's really the power. So much of the work that I'm sure other guests have talked about all relies on these tools. And so understanding those tools is pretty important, and yet there is quite a lot that we still don't understand. And this is what happens in the reprogramming field with the AAVs, which are, you pick up a neuroscience paper today, it's quite hard to avoid AAVs because people are using them to express calcium indicators or optogenetic actuators or just fluorescent proteins or use special, tracers to trace, connections. But it turns out that actually there's stuff that we still don't understand.
Yeah. So how do you overcome that problem in your research?
Yeah, I mean, so we used a different type of virus. So I think one thing to say is, so I've actually done an experiment where I've taken one of those AAVs with the reprogramming factors and the red fluorescent protein, and I've injected into the cortex of a mouse where I've created a craniotomy so I can see the cortex and put some glass over the top, and then I can watch live what's happening in the mouse over time to see what's happening. And what happens is the astrocytes fail, in the adult mouse, they fail to convert into neurons and they just die. And behind them, there are neurons which take up the red fluorescent protein and they are visible.
Okay.
So I think that's using those methods that method was never used in these early experiments. And so I think using a variety of different methods to interpret what's happening is critical. So, but I think the way that we circumvent this problem is by using a different type of virus which acts really entirely differently. So it's a retrovirus. So AAVs don't integrate into the genome. They will just infect the cell, go into the nucleus. They'll sit around. They concatemerize and they sit in the nucleus and they what we call episomal, and they're just expressed from there. We use a virus called a retrovirus, which is an integrating virus. So it'll insert the viral DNA into the host genome. And it also has this really useful property which we exploit, which is that it will only do that if the cell is proliferating. And because neurons don't proliferate, only our astrocytes get targeted. And then we've characterized this in a variety of different ways to show that we are definitely doing that. And I think one of the critical ways that really convinced me that these really do come from astrocytes. I did an experiment where we use a mouse line where we've labeled all of the astrocytes green. And then we inject our virus, which is a red fluorescent reporter, in it afterwards. And then when we when we look at the cells later, we can see that the neurons have also expressed the green label from the astrocytes. So we can see that they've come from astrocytes. And now we're also, actually today, showing, I've just seen on the scope, that we can also do the same experiment where we make a window in the cortex and I can watch these astrocytes convert into neurons live, which is...
That's
incredible.
So cool,
Yeah.
It's the best proof.
So earlier on you mentioned that the experiments you do are in situ, and so I wanted to ask a bit more of the practicality of how do you do that? Like, how do you control the certain number of astrocytes that you can then target and convert into neurons? Like, do you run the risk of maybe converting like half of the astrocytes in the brain? And, you know, all of the regulatory important roles that astrocytes have, how do you make sure you have that balance? Because, of course, they're important. It's not just converting them into neurons. You also need astrocytes. So what are the practicalities and how do you manage the risks?
Yeah, I think this is going to be one of the big limitations or challenges for the field. So you need astrocytes. So as I was saying to you before, so our viruses target proliferating cells, and so therefore we need the cells to be proliferating. But I think there's also some biology here that I think the state of cellular proliferation is critical to cellular plasticity, and it's probably the case that proliferation is important biologically to why the astrocytes are able to reprogram and why the AAVs don't work in the adult where the astrocytes aren't proliferating, because proliferation is probably very key to cellular plasticity. That's the first thing. And then, so we have two models in the lab. We work in these two models where we have proliferating astrocytes and OPCs. So one of them is in the early postnatal cortex of mice. So when mice are first born, for the first week or so, even 2 weeks, probably more than a week, but into the second week, their astrocytes are still proliferating, and so are the OPCs. So we can target them then, and we can convert them then into neurons. And that's what I work with. That's the model which I work. And then the other model is we can create injuries to the brain. So in response to injury, actually in the whole body, not just the brain, there are probably conserved programs that cells enact when they are injured. And one of the consequences that often that leads to proliferation, not always, depends on the cell. And so in the brain, if you have an injury, astrocytes and OPCs proliferate. Mostly OPCs, astrocytes proliferate, but less so. But we can target them there with our retrovirus and we can convert them in the adult brain, but only in the context of after an acute injury, in the few days or weeks after an acute injury. So the reason why that's important to your question is that I've shown also in the work that I've done that when we are reprogramming, we are not depleting the cortex of astrocytes. They are because there's proliferation ongoing, we make new neurons, but also the astrocytes get replaced.
Okay, yeah. But is that like a reactive proliferation by the astrocytes, if you get what I mean? Like, is it because you're making them turn into neurons that they feel like they're being injured and then they start proliferating?
It might be, but in the context where we work, astrogenesis is happening anyway. So I mean, they're proliferating anyway.
Like young mice. Yeah.
And I think there are probably developmental mechanisms that make sure that the astrocyte, because astrocytes, I don't know if you look at a brain and you stain all the astrocytes, what you'll see is that they cover every inch. Like they fill up all the space, like their processes, they're very uniformly spaced and their processes fill up all the gaps. It's like a tiling, almost like a mosaic. And so I think there are probably developmental mechanisms in the developing cortex in early life that make sure that you have this proper tiling of astrocytes so that even though we're taking some out, they're being replaced. And I don't think it's probably not because of an injury environment. But it is true also that there might be some reactive or injury following our injection or following the context of having to reprogram that then also stimulates their proliferation as well. It might be true. And in the injury, certainly that's what happens. So some reactive astrocytes do proliferate.
I also wanted to ask, we've touched on this briefly, but I wanted to dig deeper into the issue with, okay, let's say everything works, you convert astrocytes into neurons, and then what in terms of their connectivity? Because as we've mentioned, one of the most incredible things about the brain is how everything is connected, the wiring, the networks. And so how do you make sure you just don't have like a lump of neurons that aren't really useful and doing anything?
Yeah, so that's one of the big questions for the field. So what we know very little about this. So this is really where the field's at. There are lots of different challenges on different fronts, and I think understanding the integration is one of the ones we're at now. So what I've shown is that, so doing the same experiment with a cranial window where I take off some of the skull and I put a glass window, by expressing a calcium indicator so I can follow neuronal activity, I've been able to show that the cells, the neurons that we create from astrocytes fire action potential. I mean, I see calcium activity, so I don't know about action potentials, but they show neuronal-like calcium activity in vivo in the awake mouse. So to some extent, they are doing something, but I don't know to what extent they are really responding to the environment so we're really at the beginning of that. What we do know from some collaborators who published the paper a year or two ago is that they did so they did astrocyte to neuron conversion in the hippocampus, and they were able to show using some optogenetics where they stimulated these neurons, they were able to find, using electrophysiology by patching cells nearby, they were able to stimulate our reprogrammed neurons and detect inhibitory currents in the postsynaptic cell which they were patching from. So we know that they form connections they can stimulate or they can inhibit if it's an inhibitory neuron. And then also we can measure excitatory and inhibitory spontaneous postsynaptic potentials in the cells themselves. So we know that they are receiving some degree of input. Then I guess the other thing they did in that paper was they did this in a mouse model of epilepsy. So I didn't explain the specific subtypes of neurons we're trying to make are inhibitory ones. So they tried to do this in a mouse model of epilepsy, which was focused around the hippocampus being damaged, and that's why it has seizures. And they reprogrammed astrocytes into neurons, and they were able to show that dampened down seizure activity in the hippocampus.
Interesting. I'd be really curious to see some experiments where maybe they did like in vivo behavior and could see whether or not this would be, for example, like a place cell or a grid cell and have that kind of firing. Yeah,
that would be so cool.
Yeah. Because one thing is it like responds synaptically and it's connected. Another is it, like you said, it's actually responding to the environment.
To like sensory information.
Yeah. So this is what I'm gonna do in a month or two's time. I mean, maybe not exactly the same as a place cell, but I've shown recently that we can reprogram. So we usually work in somatosensory cortex, but now I've shown that we can reprogram in visual cortex. So we're going to do some experiments where we just look at calcium activity but show the mice some drifting gratings. And the purpose of that, by showing them visual stimuli, we'll be able to work out whether our cells are responding to the environment, which I think is really good evidence that at least they are integrated and responsive to something that's happening in the outside world. But I think it's important to realise that these cells are not the same as endogenous neurons. So
yeah, I was actually going to ask about that. Like so is there a difference between like native neurons and converted neurons and like what are the long-term differences, I guess?
Yeah, and this is one of the other big challenges or like where the field is at the moment. So we make neurons and we know that they express things that look neuronal, and we know that they can be functional to some extent, that we know that, but they are not transcriptomically, so like molecularly, they do not represent the same as any particular cell type in the cortex. So they're not becoming fully mature specific cells. They also die, so their survival is limited. So they probably end up being, for reasons that we don't understand now, they probably fail to survive in the central nervous system. And that might be to do with the immune system, it might be not wanting to reject these cells, it might be to do with the fact that they don't integrate enough. They need more activity, or it might be a hostile environment for any number of reasons. So I think that's critical. So like understanding those factors will help us to understand how to move forward with neuroregeneration in general.
I was also reading your excellent review on this topic, and where you spoke about epigenetics and how basically there's like an epigenetic conflict because you can't fully convert all the epigenetics of one cell type into that of another cell type, and there may be basically lingering epigenetic features that lead to like a metastable cell type that could potentially revert to its original identity. Have you ever seen like this reversion to original identity, or do they just die?
Yeah, so the short answer is no. Not every cell converts. But until now, I'm the first person really watching these cells live change. Until now, the way that the science usually proceeded was you would do an injection and then you would look at the brain at specific single time points in the future. And so you would see neurons and you would see some astrocytes, but what you wouldn't know is what happens between the time when it was originally an astrocyte and the astrocyte at the end. It could be that they go through some neurogenic fate and then revert back to astrocyte identity, or they could just die. So there's many different trajectories these cells might take. And so we're trying to dissect that in a variety of ways, trying to watch them over time to see how they behave in terms of morphology and function with calcium, but also we're doing in vivo single cell RNA sequencing, so we can try to plot using pseudo time. I don't know. This is not a transcriptomic podcast. It's a neuroscience podcast.
But yeah, please explain.
So in transcriptomics, one exciting thing is by what's called RNA velocity. So you can look at the ratio of so when genes are transcribed, they get spliced afterwards to form the final gene product because there are introns in most of the genes. So they get spliced out and you get your final transcript. But by someone very clever figured out that if you look at by looking at the ratio of spliced and unspliced genes, you can kind of work out which transcriptomic trajectory a cell is taking through space, like where it might go next in terms of its gene expression. And so by doing that, we can try to figure out the paths that cells take, whether they go towards a neural fate, whether they go to, or which type of neural fate they're going towards, or if they're going to a death state, or if they're coming back around to just remain in an astrocyte state. So using those different approaches, we try to figure out more about the journeys that cells are taking.
That's so cool. Wait, what is it called? Pseudo...
So, I mean, at the end of it, you get a pseudo-time plot. So you can see, it's like, because it's not real time, it's inferred. You do a lot of different time points, then you infer the gaps, basically, by... Or you can even do it in one time plot, but you have different types of cells, and you assume that cells are going to move through this space. So you might have clusters of astrocyte-like transcription and neuron transcription, but by looking at some of the astrocytes might have some early signs they might be becoming a neuron, so they would be ahead in time, but it's not real time, it's pseudo time, moving from an astrocyte to a neuron.
And what does this actually look like? Like, can you see like the genetic code, or what do you...
So you're looking at RNA,
Okay.
So it's mRNA, so it's transcribed genes. But you're looking over 20,000 genes, so you get a lot of information about a cell, about all the different levels of all the genes it's expressing. So you can see if it's just about to start expressing neuronal genes, then you might say that's moving towards a neuronal fate. Yeah.
Cool. Yeah.
So we're using these RNA-seq to try to understand more about the journey which cells might take and whether there might be these metastable states. But I think the interesting thing that you were mentioning about epigenetic conflict is that one thing I think we have some evidence, at least in vitro, not in vivo, is that these cells don't completely eradicate their astrocyte identity. So we're using transcription factors which essentially force the expression of neuronal genes and force them towards a neuronal state. But the genome is very complex and very difficult to organize, and it looks like a lot of the marks, epigenetic marks in the cell, still maintain that astrocyte-like identity. So these cells are confused, is what I'm saying. They want to be neurons, and they also still have some marks of astrocytes, and also some of the aging marks. So one of the applications of direct reprogramming for experimentalists is when you use iPSC technology to make neurons in a dish. For example, you might want to study, I don't know, whatever cells or neurons you're interested in studying for your, if you're interested in Parkinson's, you might want to make midbrain dopaminergic neurons in a dish. But you've erased, if you're doing neurodegenerative research, so if you're interested in like Alzheimer's or Parkinson's, any illness, neurological illness in later life, you've erased, to get those neurons, you've already erased all the epigenetic marks that led to those cells being old. And so one of the things that direct reprogramming can do is by preserving all those epigenetic marks, or at least preserving them better, gives you a better sense of an aged neuron in a dish. So people are interested in that aspect of direct reprogramming, not for in vivo regeneration, but just also for studying the effects of aging.
That's cool, yeah, that makes sense.
But also, like, you know, with Alzheimer's and other neurodegenerative diseases, if you have the ability to regenerate the neurons to a point where it doesn't have any of the aging markers, any of the like misfolded proteins or anything like that, why not just do that as opposed to like completely?
So that's a great. So people ask. So I was talking about the Yamanaka factors. So like the factors that can convert a somatic cell back to an stem cell, an induced pluripotent stem cell. Those factors people have overexpressed in neurons in the retina, for example, and they showed that they could rejuvenate those cells, which restored vision in a mouse model of glaucoma. So people are interested in this epigenetic reprogramming of cells as a therapy, full stop.
Okay.
So, and I don't think it's not like one of these is the best. It depends on the pathology. It's always going to depend on the pathology, exactly what you're trying to achieve. So like in some cases, if all the neurons die, you can't do that because they're all gone. So you need to replace them. But if you've got neurons there which are maybe degenerating or damaged, and you can rejuvenate them, that's a cool idea.
Okay, nice.
I also love that idea for neurodevelopmental disorders, because I think one of the things about common neurodevelopmental disorders like autism and psychotic disorders, so it looks like these are things that happen during development, can be quite early, but it can also happen during sort of after we're born, so in sort of later neurodevelopment. And you might end up with circuit abnormalities where if you were able to rejuvenate the neurons and induce some plasticity to the neurons, which would allow them to maybe make new synaptic partners or anything like that, you might provide a window of opportunity to correct some circuit abnormalities. And so that could also be a treatment for some of the more ingrained neurodevelopmental disorders that we currently can't treat at all.
Amazing.
That kind of, like the idea of opening windows of plasticity, which I love. It also reminded me of this general approach, which like our latest episode was on bio-inspired and biomimetic materials, which seems like something completely different. But I was just thinking about how I like the idea of like, you know, playing with the system and inducing some change, but also being able to understand the plasticity, the natural plasticity and homeostatic responses that occur enough to like use it to our advantage as well. Like manipulate the system to a certain extent, but then allow the natural plasticity to do its own work to let the neurons integrate into the circuits.
Yeah.
And yeah, just wanted to mention that. I also just was wondering, like you mentioned that there's two different time points or models to study this, and one is like in younger mice where there's proliferating cells, and one is when there's injury. So are the neurons that are formed from glia in these two scenarios significantly different?
I basically, I don't know the answer to that. But we don't know the answer to that yet.
Okay.
We need to do in vivo RNA-seq, transcriptomically characterize them. We also need to do 2-photon calcium, like all the functional screens I was telling you about that I'm doing in the pup. We need to do them in the adult. So I mean, these are current questions. Essentially, we don't really know.
Okay.
I mean, in the end, these neurons are immature and probably have conflicts of whether they want to be a neuron. So I think we're still at a point where I think we need to really figure out how to get these guys thriving and surviving.
Surviving and thriving.
Surviving and thriving.
Amazing. Going back to the topic on epigenetics and about whether the neuron is confused or the cell is confused whether it's an astrocyte or a neuron, could that potentially be an advantage, like this hybrid astrocyte neuron that can, you know, have all of the amazing regulatory functions of an astrocyte but also have all of the computational power of a neuron?
I mean, that's really cool. Great. I mean, another great question, which I don't think anyone else is like, can a cell, is there an infinite space in which cells can exist, or are cells always going to be pigeonholed into certain stable transcriptomic or like functional? If you imagine like a high dimensional space of everything it's expressing and everything that's going on in the cell and all of its interactions and stuff, can cells really can we make anything we want?
But
or is
that's actually a really good point that has to be mentioned, is what is cellular identity, which is really what we should have started with. Because so far we've been speaking about it like it's just the genes that a cell expresses, which determines its function also. But like, because when you said that about how many like identities could a cell have, right? So if you think about the cell type that has the most identities per se is like a pluripotent stem cell, but that's based on its potential to become anything, not on its like current state.
We like to characterize things in biology, and so like there's excitatory neurons and inhibitory neurons, and there's like all these different types of inhibitory neurons which have their own characteristics, and we like to categorize. Like the way I guess I like to think about identity is that if you imagine some hypothetical space where we have all, very high dimensional space where we have everything we could know about the cell is known, like everything that it's expressing, all of the different orientation of its genome, who it's connected to, just how it behaves, like its electrophysiology or it's like everything. That is what we where that point is in its space, in that high dimensional space, is kind of what we mean by identity. But of course we cluster things that are quite similar, so they might have similarities in transcriptomics or similarities in function or similarities in structure, and say, okay, these guys all look kind of similar, therefore we're going to call them all this cell type. And sometimes that's clear and sometimes that's not clear, and sometimes it's hard to know. Sometimes people make discoveries and then suddenly a big cluster splits into two, and so you end up with this zoo of different cellular, you can't see my air quotes, but
quote unquote identities.
But also when you were saying, like, if we characterize it based on transcriptome and the electrophysiology, if you think about it, if you characterize it to the maximum, every single individual cell could be probably its own cell type.
But it is
because there's so much variety,
exactly.
right? Even within what we think of as a cell type.
That's how I think about it. So we group them together for useful biological and scientific, and being able to communicate with each other and study stuff. And of course they do share similarities, but ultimately a cell has its own being and it has its own life and it's its own thing.
And I also think, it relates also to function. And so if we have a cell that has some astrocytic epigenetics and some neuronal ones, maybe the fact that it has this specific structure means that it can have a function where it can behave as both.
But and that's why it's pretty remarkable that some studies have shown that this conversion, maybe you're not making perfect neurons that last ages, but it does have a clinical outcome. And as we know, a lot of clinical solutions are actually not maybe you'll disagree with me, but they're not necessarily the most scientifically sound. They're just the one that works functionally. It doesn't mean that the biology has to be perfect.
Yeah, I guess at the end of the day with medicine, you balance risks and benefit, right? So I mean, if someone's getting some benefit and the risk is low enough for that benefit, then we use it. And often we don't even know why it works. I mean, there's so many examples of...
I think that's what Clara was getting at, in that a lot of the times with medicine, we don't know how it works, why it works, but it just works, so we keep on prescribing it.
Yeah. I mean, I think it's also important to bear in mind that we're really at the beginning of this journey of thinking about how to regenerate. I mean, it started right back with Ramon y Cajal and his hypotheses about
everything started.
But we still are grappling and we're making progress, but we have a huge way to go in terms of regenerating the cortex. It's very stubbornly it's not an environment that wants to be regenerated. I mean, one of the classic experiments, probably from 40 years ago, maybe more, is if you cut one of the neurons from the dorsal root ganglion in the spinal cord that goes out to the periphery, it will regenerate. But if you cut one of the ones that go up centrally up to the cortex, it won't regenerate. However, if you transect one of the ones in the periphery and then you wait a few days and then you transect the central one, that central one does attempt to regenerate. So cells can be made to regenerate, but there seems to be something about the central nervous system, which in the mammals, that really it doesn't want to let us do the job. And of course, another thing about that is that, of course, in lots of other species like fish and amphibians, if you just damage their nervous system, they'll just regenerate it. And in our periphery. So, I mean, it's like trying to unlock those secrets of biology to restore our central nervous system feels like something which biology has done before, and we can see examples of it. But for some reason, our central nervous system doesn't want to do that naturally, and we have to try to figure out why. But we're quite early on.
Sounds like it. I will never understand why the brain is so fragile, being so important for humans.
Yeah, and something like regeneration that's so important and is probably conserved across many species. It's interesting why not on human beings.
Yeah. I think that's enough about cellular identity and reprogramming for today. Thank you so much, Sydney, for sharing your wisdom.
No worries. Thanks
It's
for
been a
having
pleasure.
me.
And yeah, thank you so much for listening to today's episode. We hope you enjoyed as well. And do check out our website, neuroversepod.com, for all resources to the papers we've mentioned in today's episode.
And if you enjoyed today's episode, please feel free to share it on Instagram, Twitter, neuroverse_pod, and let us know what your opinions are on cellular identity and regeneration.
Thank you.